First of all, Chapter 9 we do not cover in this class, so you'll be looking at your next chapter being Chapter 10, and that's the last chapter that's going to be on this next exam that is coming up. So remember, exam 2 is going to cover the remainder of 5, 6, 7, 8, and now 10. So the first part of Chapter 10 goes really fast. I'm not even going to pull the slides up for you guys because most of this is just a review on what you already know. We classify microorganisms based upon a couple of different things. We name them in a universal way so that if I'm a scientist and I'm going to Germany and I don't speak German and I say Escherichia coli, it still means the same thing to a scientist that does speak German. So we classify these organisms and we refer to their identity because that is a way that we can keep a record of all of the history and species of microbes that we have here on Earth. Many people have contributed in even as I think it was 2009, I think it was. But my point is, from back in the 1800s, still to current day, we are contributing to what we know about microbiology. We do list microbes in the prokaryotes. Remember, we have prokaryotes and eukaryotes. The prokaryotic species have bacteria and archaea. You guys know the difference between bacteria and archaea. You know about their cell wall. You know about how the bacteria and archaea do binary fission, whereas the eukaryotes do their growth by mitosis. We've talked before about the endosymbiotic theory, and based upon just findings from the ribosome count, we believe that the mitochondria maybe had once been uptaken and instead of broken down was actually utilized for ATP production. We also talked about how to write scientific names. You guys know that scientific names are based upon either who founded them or a special characteristic about them, maybe where they're found in the body. And you guys also know that when you write them out fully the first time, both genus and species. The genus is capitalized and the genus and species both are in italics. We do know that we define species much different between a eukaryote and a prokaryote. For eukaryotes, we define those species based upon a group of animals that are closely related, and they're going to be organisms that are breeding amongst themselves. You don't see a rabbit and an elephant breeding. There's a reason for that. So eukaryotic species, much different in how we define them as opposed to prokaryotes. With prokaryotes, we define them based upon a population of cells that have characteristics that are very similar. So we can have a clone. If you have a population that was derived from just one cell, so you started off as one cell, that cell copied its DNA and underwent binary fission, and now you have multiple cells, that's a clone. All of the cells in there are going to be exactly identical. Or you can have a strain. A strain is when you have genetically different cells within a clone. So maybe it's a culture, but they're not identical. Maybe there's been some mutations. Maybe there's been conjugation. We talked about those in Chapter 8. When we look at viral species, we typically base viruses and group those together on the signs and symptoms that they cause, and then on which part of the body that they tend to infect. For example, if I tell you that I have a respiratory virus and I'm very sick, signs and symptoms that you would automatically think of would not be diarrhea and vomiting. That's a GI virus. Signs and symptoms of respiratory viruses are things that are going to be like cough, sneezing, sore throat, upper airway type of junk. When we talk about the difference in eukaryotes and prokaryotes. You guys already know that for the most part we're going to focus this class on prokaryotic species, and we're going to take a look specifically after chapter 10 at a more detailed look at some actual types of bacteria. And then we're going to take a look at some of the eukaryotes, and we're going to talk about mold and mildew and yeast, and we're going to talk about algae, and we're going to talk about helminths and protists, and then we're going to move forward and we're going to talk about viruses, and we're going to talk about some individual species and what they cause. There is a reference manual that is actually called the Bergey's Manual, and this is usually in several of the labs that we have pretty much anywhere. And what it is, it's an identification or a reference for help with identification of microbes. Because one of the things that we have to do in a lab, if we're going to have a species and we're going to have it as an unknown species, the question is, how to identify it? How do I know exactly what I am working with? Well, for the most part, with prokaryotes, we know the first step is going to be to do a gram stain. You gram stain it for me and you tell me, is it a gram positive or is it a gram negative? But that's not the end of the story. What we do in a lab is we have what's called a dichotomous key. So with the dichotomous key, we're going to start up here at the top and we're going to do a test. And it's a test that we can answer yes or no. So maybe this test is going to be, is it a fermenter? You guys know what the fermentation test is. And so we actually have to perform that test. We do the fermentation test and then we say, okay, is it a fermenter? If the answer is yes, then we come over here to this box, right? And we perform this test. And maybe that test is, okay, is it oxidase positive? That's a different type of test. And essentially this key allows us to work our way down into, sometimes it's multiple layers of work. But what it does, it allows us to peel back the different sections and to identify that microbe. So I know this first introductory to chapter 10 was very short. The next part of chapter 10 is going to look at some of those testing that we actually do in order to identify microbes. But it's very important that you keep note of what your goal is. Your goal is to be able to identify a microbe that you have no idea what it is. Patient comes in and has a sample and someone says, we have no clue what this is. You figure it out. You've got to be able to do that. And so these tests are a way that we are able to take unknown species and figure out what we have. So that's where we're going to go in chapter 10. I'm going to leave you there for tonight, and we will talk tomorrow on some of those specific tests. Hope you have a good evening. See you later.